way to prove me wrong. So evolution needs this if-then hypothesis. And after a few decades of people talking about evolution, a couple of scientists, a mathematician and a medical doctor, one from England and one from Germany, named Hardy and Weinberg, came up with the Hardy-Weinberg law of equilibrium. And it's a mathematical proof. And it is essentially what it is the null hypothesis. And what that means is it is saying, no, evolution is not happening. If a population of interbreeding organisms comes up with this mathematical outcome, then evolution is not happening, right? So this is providing the null hypothesis. This is providing the no. You can't just say evolution is happening, end of story. But what Hardy and Weinberg came up with was the scientific way out to say, no, actually evolution is wrong if these things occur in this way. Now, if you look at your book and you see the Hardy-Weinberg test for evolution, the law of equilibrium, it may look a little bit daunting because it's p squared plus 2pq plus q squared equals 1. Don't be frightened by this. This is actually very simple and we've seen it already. When we looked at Gregor Mendel's pea plants, remember that? We had a tall T and a small t. So you had the tall plants and the short plants. What Hardy and Weinberg are doing there is they're generalizing that principle. They're not just looking at one trait of from pea plants. They're saying traits in general can be distilled into a mathematical ideal. And so your book actually has, they don't use capital T's and lowercase t's. In this case, they're using capital A's and lowercase a's. But it's the same thing, right? You have two alleles. You have a tall plant, a short plant, or this characteristic and this other allele, this other variant. And in this case, A is the dominant, and capital A is the dominant, and lowercase a is the recessive allele. Now, what Hardy and Weinberg are doing is they're replacing these individual characteristics with the principle of a characteristic. And so P is the replacement for the uppercase A, right? And that would be A times A, which is the purebred uppercase A. And you have the Q, which is represented for the lowercase a, and that's also squared, so lowercase a, lowercase a. And if you add the P's and the Q's and 2PQ together, you will equal 1, which is 100% of the population. Now, the use of this this formula is important because what you can then do is you're not just testing a hypothetical population of interbreeding organisms. I can go out into some forest someplace and I can make observations of a genetic population that's interbreeding, and I can plug not just 25%, 25%, and 50% into these things. I can plug the real numbers into them, a real life situation. And if one generation to the next you have a maintenance of the same percentages, then Hardy-Weinberg are saying if that's the case, if the percentages don't change, then evolution is not taking place. That's fine. Hardy-Weinberg were not trying to refute evolution. What they were trying to do was to provide some explanatory added value to evolution. So we don't come across Hardy-Weinberg equilibrium very frequently because populations are constantly changing. But what's more important about this Hardy-Weinberg test for evolution is that once you've plug in your real values of different characteristics into that equation, you can tell not just whether evolution is taking place, but how it's taking place. Is it moving in this direction, that this particular allele is being selected by nature, or is it moving in another direction where this population is losing that feature? So you can actually see the directionality of evolution through this. It's not just whether evolution is taking place, but how evolution is taking place, and that's really important. So let's go to some of these principles, and I'm going to just remind you again, and when I remind you multiple times about things and I give you vocabulary, take it as a point that you probably should learn this stuff. What are the 4 mechanisms of evolution? We've already looked at, Darwin told us about natural selection, and then Gregor Mendel brought in genetics, which have 3 factors that are evolutionary mechanisms. That's mutation, gene flow, and genetic drift. So let's look at those things and how natural selection is going to be sort of a little bit more nebulous, because each that's constantly changing. It's a constantly changing variable. But the things that are genetic are kind of principles that guide genetic evolution. So mutation is the only place where new alleles are added. We looked last chapter that there's a huge variety for natural selection to select from based on just recombination of things, right? So just the simple process of sexual reproduction produces a huge variety of in the next generation that natural selection can select from. But even on top of that, mutation allows for even more variety, and something that nature has never seen before, that genetics have never seen before. But what are the forms of mutations? Well, we looked at the structure of DNA is actually just a sequence of chemicals. I had the metaphor that they were like magnets, right, that come together. You will never get a G with a T. You'll never get guanine with thymine. That can't happen. They repel one another. Whereas adenine and thymine attract one another. You always get an A with a T. So you're never going to have that. That's not a mutation. That can't happen. But what you can have is you can have them flip-flopped. So you can have where these are called point mutations, that the chemical in that sequence can be actually exchanged. So you can have where a T was here, you have them actually flip-flopped, and you've got an A here and a T here, where it previously was a T and an A. Now, that could be, the result could be really nothing that you'd ever notice. And in fact, if you go back to that chart that I wanted you to print out and put on your refrigerator as a good way to watch your diet, you can see some of these things. Like, for instance, for isoleucine, you can have TAA or TAT. And TAA and TAT will produce the same amino acid. So that means that the T at the end was transposed. But the outcome is the same. The same amino acid is being produced in that triplet code. So that is what we call a synonymous point mutation. But if you have something where you have a transposed element that is not synonymous, that would mean that you would produce something else, like a different amino acid. So for instance, I'm just looking at the chart right here myself for something that CCA is glycine, which works as a neurotransmitter. And let's see, CTA is aspartic acid, which promotes mineral uptake in intestinal tract, right? They do different things, and if you have a point mutation that produces something different in your body, then it can have an effect on your health, your outcome. It can be positive, it can be negative. We don't know, just by saying that it's a non-synonymous point mutation. But these are the two things, that a mutation can be something that produces the same effect, which is synonymous, or a point mutation where that particular chemical has been flip-flopped or exchanged with something else, and it's non-synonymous, producing a different effect in the body. Another type of mutation is called what's called a frame shift mutation. And this is like you've got your sequence of codes and you slip something in there where it didn't belong. And that means that everything above it is okay, but everything below it is out of order. It's like if you write a sentence in letters, right? And you write out a sentence on a piece of paper, and in between each sentence you've got breaks, right? In the words, like I am teaching Anthropology 102, right? So in between I and am and teaching, there's breaks. But if you slip a letter in there and you keep those breaks, it would be a nonsense sentence, right? After the first part, I am, but then teaching might have another letter slipped in there, and then the end of the word teaching would have a different gap in it, and the next word, anthropology, would be ganthropology or something like that, ganthropology actually, and then Y102, right? So it would be a nonsense set of letters after that extra letter was slipped into the word teaching. And this is the same thing with DNA, that if you slip something in, all of those codes after it are going to be shifted down a place, and the gaps between them and where the RNA comes to get its instructions for doing stuff is going to be out of place. And so this can have a really serious impact on the DNA structure. So a frame shift mutation is not like a point mutation. It can have a much larger effect by shifting all the nucleotides down by 1 point. Also, certain elements, we looked at how there was linkages and tying together, that you can actually swap things around, whole parts of codes to different places, and that can be either something that retains its synonymity, where it actually, you can still get that code, or it can move it out of place in such a way that it doesn't function anymore. These are called transposable elements, so big chunks of DNA can actually move around. And most mutations that we're dealing with in evolution are spontaneous, and about on average, they're absolutely unpredictable. You cannot predict when a mutation is going to occur or how it's going to occur, but that's the chance of life, right? That's the chance that evolution is giving us to do something, either to have, so it's a roll of the dice. So they're spontaneous, and on average there's about 1 new mutation per every person born. But you can also have induced mutations, which are the result of things like radiation. Even the sunlight mutates your skin, which is where skin cancer comes from, is your DNA structure in your skin cells mutating and growing cancerous. By that rays. So X-rays, toxic chemicals, radiation, these sorts of things can induce mutations and often have harmful effects on your DNA. Another form of mutation is what's called duplication. And this is when whole chromosomes, remember, chromosome is the whole stretch of DNA that's compressed into a manageable form. Sometimes whole chromosomes can be duplicated, or parts of chromosomes can be duplicated. And there's several types of chromosomal duplication effects that we know about as genetic, that have genetic consequences. So there's trisomies, and monosomies, for instance, where you are missing a whole chromosome, and there's trisomies where you have an extra chromosome. One of those trisomies is called Klinefelter syndrome, which is where you have an extra X chromosome. And people that are born with Klinefelter syndrome have 47 chromosomes as opposed to the usual 46, and that trisomy occurs on their sex chromosomes, which means that these people often have are born with both male and female characteristics, that they are actually an intersex condition. These mutations are neither good nor bad in themselves, right? Nature doesn't care whether you live or die. It's just, nature is just nature. And in fact, I hate to break to you, we all die. But the question is, are you going to see your genes passed down into the next generation? That's the only thing that is really relevant in evolution. And mutations likewise are neither good nor bad. It's situational according to natural selection. So, for instance, an albino, a non-melanic crocodile or alligator isn't going to survive in the wild very much. You see them in zoos because we feed them. But an alligator or a crocodile, they're ambush predators. They rely on being able to sneak up within striking distance to their prey that's getting a drink of water, and then they snap very quickly. They're not going to be able to do that if they're stick out in a dark pond and they're white colored. That's not very good. But having a white coat in a snowy environment as a hare, an Arctic hare, is probably going to save your life. So having non-melanic characteristics, having white pigmentation or no pigmentation as an alligator is probably going to kill you, whereas having a white coat as an Arctic hare is going to save your life, right? So there's also mutations like a striped cheetah, right? Does that make any difference? Probably not. Are they still going to be able to chase down their prey? Are they still going to be able to stalk close enough through the tall grass? Yeah, probably. Do the stripes really make a difference over spots? Probably not a huge amount. So that's something that's just benign. It's a mutation, but it doesn't make much of a difference. But also remember that every characteristic has a drain on your resources, that if you are growing something in your body, you got to pay for it through nutrition and energy, and you've got to build it. Your body is building you. And if it's not useful to you, it's a drain and a drag, and it's in aggregate going to slow the process of passing down your genes to the next generation. So if you don't need it, lose it. And nature will make sure that you do. So something like an extra eye that forms on the wing of a fly it's a mutation. Does it help? It's probably a non-functioning eye, but eyes are expensive, right? There's a lot of nutrition and energy that's put into growing an eye. And so does it help the fly? No. Does it hurt the fly? Not actively, but it is a drain on it. And so it is probably going to be less competitive than, at least over generations, from its non-eye on its wing cousins. And all of these things add up to aggregate. So at the start of in this chapter, there's a picture of a seahorse that looks very much like its surroundings. It has all of these appendages that look like kelp, and it blends into its background, and that helps to save its life. But if those conditions change, and you can see a picture of that same seahorse on the backdrop of a dark blue ocean, it's going to stand out like anything, and it's going to be food. So if those conditions change, then new adaptations need to occur. So you have a buildup of adaptations towards an environment, but if those change, then it's going to take off in a new direction. So natural selection is another one of these mechanisms of evolution, and the Hardy-Weinberg principle allows a law of equilibrium, allows us to tell the direction that nature is selecting a population of interbreeding organisms is taking. There's sort of a graphic that shows directional selection, stabilizing selection, and disruptive selection, and then no selection, which is sort of the Hardy-Weinberg principle of no evolution taking place. And that no selection requires there to be sort of a non-pattern to it, that it is that it's not pulling in one direction, because as you'll see that through time, stabilizing selection and no selection look the same, that actually something like a cockroach is in a stabilizing selective pattern, but it is directional because the margins, the largest cockroaches and the smallest cockroaches are being selected against, and it's actually the ones in terms of size that are being selected as the most successful. In no evolution, you wouldn't have those pressures on the margins. Now, here's a metaphor for the way this works, right? That take a hippopotamus, right? Now, you have a population of interbreeding hippopotami that are in the Nile Valley, or let's say the Nile Delta, and they're all frolicking around being hippopotami. And they're pretty large creatures, right? And that helps them to ward off predators, because there's a lot of dangerous predators in where they live. Crocodiles, lions, that sorts of thing. Humans even. So it helps to be large, which means that you're going to have natural selection through predation, through predators eating the smallest ones, are going to be pulling that directionally towards larger size, right? So hippopotami are going to be larger. That helps them to survive. But here's the key. How large do they get? Do they just keep getting bigger and bigger and bigger until they're larger than elephants? Well, no, because there's a limit to how much food there is available. So the smaller you are, the less you need to eat, which means that you are going to have to find less food than your larger colleague, right? So if you get too large, you'll struggle to feed yourself and you then could die, which means that there's natural selective pressure coming from the other direction, pushing this population down in size. So you've got one population, predation, pushing it up in size, and another pushing it down in size, and the middle is what's being selected. And so you have what are normal-sized hippopotami, which are pretty large but not too large and not too small, right? Great. That is the environment that hippopotami are living in. Now imagine there's a troop of hippopotami that made it down to the Mediterranean coast and were frolicking around in the sea, and a big storm comes and pushes them out to sea, and they manage to find some vegetation that they raft on, and they make it to an island. Say Crete or Cyprus, one of these Eastern Mediterranean islands. And they're like, whoo, that was lucky. We're lucky to have survived. Let's go find some food here and let's start breeding, right? Okay, they